Okay, now that we're being recorded, it's my pleasure to introduce Dr. Prem Kandel, who is a postdoc who joined my lab in late 2017. So Prem started off getting his Bachelor's of Science in Agriculture from Tribhuvan University in Nepal, where he's originally from. And then he went on to get a Master's of Science in Agriculture with an emphasis on plant protection from the Hebrew University of Jerusalem in Israel. And this was magna cum laude. And then from there, he moved on to studying plant pathology. And that's sort of his field now that he's kept with. So he got his PhD from Auburn University in a lab that works on Xylella fastidiosa. And so for people who don't know, Xylella is a real pain to work with because it's fastidious. It takes a long time to grow. And so he did both some genomic work and some molecular work. So he's very good and has a lot of patience to be able to pull that off. And so with that, he'll be talking today about some of the different stressors that my lab is interested in and that he is interested in as well, and sort of how bacteria can tolerate those stressors. Yeah, you're muted, Prem.
Yeah, okay. Am I good? Okay, yeah, thank you. Thank you for that nice introduction. And it's my pleasure to have this opportunity to talk. And thank you all for joining. So I'll be talking about my talk is divided into three major components. In the first part, I will talk about little background. Then I will move on to Tailocin persistence and then to antibiotic persistence. So this work is done in a model plant pathogenic bacterium, which is Pseudomonas syringae. So Pseudomonas syringae is a gram-negative bacterium that has been isolated from almost any types of environments, but when conditions are favorable, it causes serious diseases in plants. So this is actually a model bacterium to study plant bacterial interactions where a lot of the virulence factors and the plant defense mechanisms were studied first. That means that it has a lot of tools to work with, but genetic tools and others. And strains are grouped into pathovars depending on their host strains and some disease characteristics. And there are more than 60 different pathovars described. Some here are diseases of two pathovars, Pseudomonas syringae pathovar syringae PSY and Pseudomonas syringae pathovar Fasciola PPH. So we can see that both of these pathovars infect green beans, and our lab is actually interested to understand this interaction between the pathovars when they co-infect the same host. But green beans are not the only plants that are infected by Pseudomonas syringae. There are many other economically important crops. Carol was talking about tomatoes, so it's a pathogen of tomato too, a kiwi fruit, and all others. So as plant pathologists, we really get excited when we see these nice symptoms in our field trials or greenhouse trials. But when growers have these symptoms, that is actually a real problem because they minimize both the yield and quality of their crops. But the sad thing is that we do not have good control options to control these diseases. There are some copper-based bactericides or some antibiotics that have been prescribed, but they are very restricted to be used in agriculture, especially plant agriculture, as these statistics suggest only almost 0.1% is used in plant agriculture compared to animal agriculture. So even if they were to be used, there are problems associated obviously with the resistance evolution. So these treatments will fail pretty fast. And this might be of interest to the microbiome people, obviously, because bactericides are usually broad spectrum. So they kill not just the pathogens, but almost everything, even the beneficial microbes, which can again have negative consequences. So they suggest that we need alternative ways to control these pathogens. And one of the alternatives that have been proposed for some time are bacteriocenes. So as this review title points out, can bacteriocenes be viable alternatives to antibiotics is the question, my research is trying to address to some extent. So before I get into my research, just want to introduce what bacteriocenes are. So bacteriocenes are protein toxins that are produced by bacteria. So the bacteria have genes to encode these toxins. So they are narrow spectrum. They usually target closely related strains. So they are known to be anti-competitor compounds. So there are many different classes of bacteriocenes, but tailocins are bacteriocenes that resemble bacteriophage tails. They are also known as phage tail-like bacteriocenes, as we see this structure. So when the producing cell is induced, this makes these tailocin toxins, and to release these toxins actually lies the producing cell. So this is actually a high cost for the producing cell to release these toxins. So these are found to be potent killers in a closely related Pseudomonas aeruginosa. And when they are released in the environment, they go on to bind to these target cell receptors, which are known to be lipopolysaccharides, these outermost component of the gram-negative cell wall. They puncture through the cell membrane and cause membrane depolarization and cell death. So in Pseudomonas syringae, the work by my postdoc mentor, Dr. Kevin Hockett, saw that this PSY B728A produces this tailocin, which can actually kill a lot of these pathovirus, some of the disease symptoms I saw already. And I have this picture here where these purified tailocin particles, when diluted up to 1,000 times, still show killing activity to one of these target PPHs. So the PPH target strains are growing in that lawn, and then when we spot these tailocin supernatants, we see those clearing zones. That means that they are inhibiting or killing these target strains. So this is from the in vitro data, but not only in the in vitro conditions, also in the in planta conditions. The work done by our PhD candidate Honoria has shown that when the producing strain is able to produce active tailocin particles, they actually antagonize the sensitive strain, as you see this difference in the population. Remember that the producing strain has to lyse themselves to release these toxins, and even then so there's not a lot of toxin being produced in the in planta conditions, but even then we see this effect suggesting that they are actually highly efficient in the in planta environment. And not only this, a very recent preprint, actually it was published maybe 2 weeks before from the Deb Baltus lab, in which Dr. Hockett is also an co-author, showed that when tailocins are applied as pre-treatment, they significantly reduced the pathogen population in the plants. So in the y-axis, we have this pathogen population. On the x-axis, we have these different tailocin supernatants. Some of them are active and some of them are not active. So when the tailocins are active, we see that the pathogen population is significantly reduced compared to when there's inactive tailocins. And when that is actually fixed, the defect to produce active tailocin is fixed, we again get that low population in the plants. And this picture also shows that these pathogen uninfected plants look pretty healthy, and these all others were infected with pathogens, but these two had active tailocins applied. So we can see that when the active tailocin is applied, we see no disease. But when there is no active tailocin, but only pathogen, we actually see no plants. So this kind of clearly suggests that tailocins are actually very good in controlling pathogens in the in-planta environments too. So when we get back to that question again, yes, they can be good alternatives, but can they really be viable is the question that I'm interested in, mainly in terms of if these pathogens have mechanisms to avoid killing by these tailocins. And when it comes to avoidance of killing, we are very familiar with the resistance evolution, especially with antibiotics. This occurs by genetic mutation in the antimicrobial target. So another mechanism that enables survival to antimicrobials is persistence. This is known in clinical pathogens a lot, but this has been increasingly recognized in the plant pathogens too. So what happens in persistence is that there is this pathogen population when we apply antimicrobials. There are some cells that are not killed. All the sensitive cells are killed, but these persister cells are not killed. And then since they do not have a genetic mutation, they are caused by some physiological changes. So when the antimicrobial stress is gone, they actually give rise to this wild type population. So I mentioned that resistance is caused by genetic mutation in the antimicrobial target, and persistence can be caused by a number of different mechanisms. The well-known mechanisms are toxin antitoxin systems. So there are at least 4 different types of TA systems, and the type 2 TA systems are well known. So I have this cartoon showing what these type 2 TA systems are and what they do. So these are 2 gene modules. They occur in an operon. So the antitoxin component encodes this antitoxin protein. Toxin component encodes toxin protein. So under a normal condition of the cell, the antitoxin tightly binds to this toxin. So that inactivates the toxin molecule. But when the cell is under stress, some proteases degrade this antitoxin component, freeing that toxin. So now the toxin is active and it acts on a number of different processes of the cell, such as it degrades mRNA or inhibits protein synthesis, or it also inhibits replication. So that makes these stressed cell kind of dormant, and they do not respond to any of the antimicrobials that we apply. So my goal here is to understand these mechanisms, if there's like a TA system, so some other mechanisms that might have a role in tailocin survival. But first thing first, about the resistance, we have isolated a number of resistant mutants. When we expose these cells to tailocin, we have sequenced their genomes and found that mutation in the LPS biogenesis genes actually causes tailocin resistance. As we have in this picture, so these resistant mutants actually lose their O antigen, LPS O antigen. Know that the LPS consists of 3 different components: the O antigen, core, oligosaccharides, and lipid A. And when cells get tailocin resistance, they lose this O antigen. As we can see that these do not have the O antigen band. And when that happens, they actually lose their host fitness. As we can see here, the in-planta population is significantly reduced for these tailocin resistant mutants. That is actually equivalent to a type 3 secretion mutant. So this suggests that resistance evolution, because they are weak in the in-planta conditions, maybe it's not much of a big concern when we apply tailocins as antimicrobials. So the next thing is, again, with persistence. So the assay to find out if there are persistent cells is to do a death curve. So that's what I have done here. So I purified these tailocin particles and then applied this lethal dose to these target cells. And what we see from here is that within the first hour of the treatment, we see a significant decrease in the population. So if the all cells were sensitive, this population will drop down to this level, even below that. But there was some slow dying cells, or these persister cells. So these curves do not go down, but kind of flatten. So these, and then when we expose them longer, I think there is like division of the resistant cells, the curve starts to grow. So this death curve suggests that there are persistent cells to tailocins, but just to confirm, we again recultured those and then re-expose them to tailocin. And what we found is that the majority of these survivors were actually persistent. And also some of them are resistant, and we actually found some incomplete resistant mutants, which I will show in the next slide. But we see that the stationary cultures survive more, and then the persistence is more in the stationary culture than in the log culture, which has been shown with antibiotic treatments too. So this is the criteria that we use to differentiate these phenotypes. So this is the short-term broth exposure assay, and this is the long-term overlay assay. In the broth, we apply these tailocin particles and monitor their reduction in population. As we can see here, for the wild type, we have this much reduction. So if this was a persistent survivor, this still is equally sensitive. It survived in the first exposure, but in re-exposure, it hasn't gained any resistance, so it is equally sensitive. And we had some high persister-like mutants that had some level of sensitivity and survived better. But in the long-term assay, we still saw equal sensitivity to wild type, and then these incomplete resistant mutants maintained some level of sensitivity, at least in one of the two settings. So this suggests that we had, and again, when I apply even a very high dose of tailocin, up to 10 times more, we still see almost a similar trend. This suggests that this tailocin persistence is real. So then we put these in the in-planta conditions, and we found that unlike the resistant mutants, which have a compromised host fitness, these persistent survivors or even the incomplete resistant mutants were equally fit in the in-planta conditions. So we now know that, so that suggests that persistence is a way to avoid killing by these toxins and also to maintain this fitness potential. So the next thing we did is to find out what may have caused, what causes this tailocin persistence. We're expecting to see some, the toxin antitoxin systems, which is not the case. So when we sequenced the genomes of these high persister-like mutants, we found mutations in these genes. And so this was one of the genes that, the hypothetical protein that we do not know the function yet, but it occurs in the same LPS operand. So we believe that it does something with the LPS modification. So there is this another gene that seems to be involved in the peptidoglycan recycling. And this was the molecular chaperone that is involved in protein folding. So what these all suggest is that there is some cell surface integrity component that is involved in persistence to tailocin. So just to recap, so we saw that tailocins are highly potent to be used as therapeutics in bacterial plant pathogens. So the tailocin resistant mutants lose their O antigen and they have a compromised host fitness. And we also saw that tailocin persistence is real, and the modification of the surface structures could lead to persistence to tailocin. So the next part is with the antibiotics. We wanted to understand what mechanisms the antibiotic persistence has in this bacterium. So we choose streptomycin as the antibiotic because it is used in agriculture too. So we can see with this death curve that persistence also occurs with streptomycin. So we aim to use streptomycin and then do a repeated exposure to select for some high persister mutants. From that, we could understand what mechanisms are involved in the antibiotic persistence. But just using streptomycin was not enough, because we got a lot of resistant cells that kind of like compromised our ability to select high persisters. So we actually used this combination treatment of streptomycin and tetracycline that actually took care of all the resistance, and we only were able to select these persister cells. So using this combination treatment, we actually did a repeated exposure, just selecting for the persister cells and then again re-exposing them to the same treatment for a total of 3 times. And then we enumerated these persister cells, and then we evaluated if they have any increased persistence. So by using this method, we have actually identified 9 different streptomycin high persister mutants. So I'm showing here their phenotype. We also had a streptomycin resistant mutant that we isolated during these experiments. And what we see here is that these resistant or the high persistent mutants grow well in the normal medium. There were few that had a reduced growth, but as soon as we apply a MIC of streptomycin, the minimum inhibitory concentration, we see that the resistant mutant grows well, but these persisters do not grow. They look equally sensitive. So this is the concentration we used, and the same thing. But when we wash these and then plate them, what we saw is that the wild type has a huge reduction in the population. They were all killed, but these high persister like mutants have almost 1,000 to even 10,000 times more survival. So what these 2 graphs suggest is that these high persister mutants did not grow at all in the presence of streptomycin, but they survived significantly better, suggesting that these are actually true high persisters. So we're really excited to see these results, so that we decided to sequence the genomes of both the resistant and the high persister lines. And we found that the resistance was caused by a mutation in this gene, which is known to be a streptomycin target in other bacteria. But the high persister mutations were caused in these 2 genes, which seem to form an operon. So the one gene is the DUF2384 domain protein. The DUF stands for domain of unknown function. The RES stands for arginine, glutamic acid, and serine domain containing proteins. We're not sure what these do, but we have found that in some other bacteria, such as Mycobacterium tuberculosis, these 2 genes have actually shown to form toxin and antitoxin systems. So we are working on the genetics of these systems, so we'll know more once we have those results. But what we have in conclusion from the second part is that persistence also occurs with not just with the Tailocins, but with antibiotics and Pseudomonas syringae. And it appears to be caused by a novel TA system. And what we saw from the Tailocin persistence and from the antibiotic persistence is that these 2 persistent mechanisms seem to be different. So the take-home message actually is persistence now is to Tailocin is real. We saw that Tailocin is effective in controlling the pathogens, but there is this persistent thing. So this actually needs to be considered when we design Tailocins as therapeutic agents. So with that, I would like to thank Dr. Kevin Hockett for his very excellent mentorship. I'd like to thank PhD candidate Honoria for sharing her data, the members of the Hockett Lab, our collaborators, Dr. David Baltus from University of Arizona, and Dr. Lindsay Triplett and Dr. Ravi Patel from the Connecticut Agriculture Experiment Station, USDA-NIFA, for funding, and the Department of Plant Pathology and Environmental Microbiology, and also the Microbiome Center for this opportunity. Thank you, and I will take any questions.